So the next step is adding data files. And there are basically two ways that we can add files to the data set. One is directly through the browser. Basically, as we can see again here in our slide, the files get linked directly to the replicate. So what we need to do is navigate down to the replicate. Let's say I want to add some sequencing files using the browser. So I will go to this experiment, navigate down to the replicate, and then in the replicate we'll see different type of data that we can add. So we can add sequencing data, process data, which are basically derivatives of processing the raw sequencing files, for example, BAM files or counts, or in the single cell data can be Seurat files. Then we have a section for the track data, which are actually the ones that get visualized in the Genome browser, which is typically in big format. Then we have imaging data, which can be very large type of 2D and 3D images. It can be in TIFF or PNG or JPEG or Nifty, a very large collection of data we support. And then we have a section for mesh data. So you have created surface data. You can load them up in there. And then we have a last section for sort of array tabular data, in case sometimes you just generate a table with data and you can upload it here. So let's say in this case I want to add some sequencing data. So I just go add record. And here, there is some information related to that type of data. Let's say I select pair n and read, for example, 1. But then the most important part here is that basically this allows you to select a file from your computer. You click select file, you navigate to a place where you might have in your local machine where you have some data. You select the file, you add some information about the file type. For example, this is a FASTQ. File. You say when you're uploading it, and click save, and this directly loads the data to the catalog. So now if you navigate back to the replicate, you will see that there's one data file that has already been uploaded. And now you can see these at different levels. So if I navigate back to the experiment, I can see that sequencing file is listed there. And if I were to navigate back to the data set, again, the same file is shown there, listed as sequencing data. So that basically shows basically a complete part of uploading data using the browser. Now, there's another mechanism that we provide for uploading data, which is when you want to upload a large number of files. You don't want to do this one by one. I mean, this mechanism with the browser is actually can go pretty fast, up to 10, 20 files. You can do it. You can actually do it very easily. But if we have some users that upload hundreds or thousands of files. So we have a tool that we call the Deriva Upload tool that allows you to organize the data in a given manner in your machine, and then install this tool in your computer and batch upload all those files in a very consistent way and very robust way. You know, this again, it's another tool that does checking to make sure that the transfer was complete and that the data is not corrupted, and also if you get disconnected, you can always go back and the upload will continue from where it left off, which is very useful for very large files. So I can talk a little bit about that. So
just for a second,
yes.
Since we've been going for a while, I just wanted to see, does anybody need a break, like a quick break, before we go into, because we're going to be doing an
it's a few more minutes. So any questions so far? Does it make sense?
Yeah, thank you. It was very detailed, and I'm glad that I had some previous information from your publication from Rob. So.
Okay. So what I wanted to show you, what I wanted to say that in your case, for example, let's say you were to, before we go into the other section, maybe we can do a little bit of a discussion about the case of how it goes into the human subject case. So let's say you were instead of being a mouse data set would have been a human subject data set, and let's say that you have some data that can be put up, like there is some completely de-identified data, and then some data that is protected data that we cannot display in here. So what we would like in those cases, what we would like to do is to basically go through the same process, create a data set, add as much information, description, study design, contributors, publication protocols, everything as much as possible, like we just did. Then create the experiments so that we know what type of how the data was obtained and what type of experiment you conducted on the data. And then same thing with the biosamples. Of course, you will only enter information that can be added. Sometimes you cannot, because you don't want to enter any. I mean, the local identifier cannot be anything that identifies the subject, of course. But or age is sometimes can also be problematic, or sex. But other things like species, genotype, strain, phenotype, or even syndromes. So that's one of the tags that we have for biosamples is syndromes, and you can add those in here.
Yeah, I think, in human samples, especially if these are some craniofacial malformations and syndrome, you do need to enter
exactly.
age and then sex, of course, because they are some,
sure.
Important characteristics which complete all that picture.
Right. So
and we don't have we have everything without identifiers, of course.
Okay.
That's IRB request anyhow, but always informations are accessible if there be anybody who needs some more information, which may also not be at the first time collected, but we can go back to the chart or database where we do have full information about that patient and the family.
Okay. Yeah. So let me show you one just very quickly. Like in the case that imagine that this will be like an actual human sample. We do have the well, I'm not sure why I'm not seeing here the I need to look into that. I don't know, but we do have a complete vocabulary with the from the Mondo ontology for the syndromes. So
I think I saw that when I was browsing through. Yeah,
Okay.
I think it is
Okay.
there. Yeah.
Okay, sounds good. Okay, so as I wanted to show you that, we basically have for phenotypes and syndromes a very large collection of proper terms from known ontologies that you can associate to your biosamples.
Yeah.
Okay, so I think that's things that I wanted to show. We can go now through the process of uploading the files using the tool. If you're interested, I don't want to tie you up if you're not. Is that something that's not interesting to you or not?
If it's okay, because looks like I'm the
Okay,
older
sure.
student.
Yes,
so
sure.
I would
Okay,
love to.
Okay, so let me go. Let me now. It's only going to take a few minutes more. So let me go back to actually continue this part in our. It's probably easier to go to some of our documentation first. So I will go to we basically have this curation wiki with a lot of information. Pretty much everything is very well detailed. So in this documentation pages, there's one section called the upload data files. And basically explains all this process very well. But the more important part that I wanted to show you is how you organize your file. Because remember, what we're going to do now is we're going to see how you can organize your files in the computer in a way that then you install this tool, point the tool to a directory, and then all your files are going to be magically uploaded to where they're supposed to go. In order to do that, what we need to do is basically look at this diagram. So basically, the way you organize the files in your computer are first you create a top-level folder that is the dataset folder, and the name of that folder has to be the record identifier of your dataset. So remember, if we go back to the let me actually get back here. If I go back to the data set that was just created, at the top of the page, there is this record ID. So that's the RID. So that is going to be the name. This 14-HQ32 is going to be the actual name of the folder. So first you will create a folder called 14-blah, blah, blah. And then under that, you will create one more folder for each of the replicates. If you remember again, here in the data set, if we look in the experiment, each replicate had this identifier. So we'll create 3 subfolders called 14-HY0Y and blah, blah, blah. Okay, so that's the next level. And then under that, we'll create subfolder for each of the different type of data. So we'll create a subfolder if we have sequencing data, and there's where we're gonna put the raw sequencing files, let's say our FASTQ files. If we have some process data, I will create a proc folder, then a subfolder for each of the mapping assembly, let's say MM10 or HG38, depending on which mapping assembly was used to create those derived data files. And over there, put all your BAM files or bigwigs or beds, or Seurat files if there's single cell data. Then for images, I will create an image subfolder and add the high resolution data in there in Nifty or OMI-TIFF or JPEG. Then we'll have the meshes, another subfolder in case we have meshes, which prefer them to be in OBJ, or they can also be in PLY format. And then if we have thumbnails for the images, we create another subfolder. And for each of the images, we create an individual folder, and we add the actual low resolution thumbnail for that. So this is basically what I wanted to take a look. And then, so I think we're ready to install the client and upload the file. So in this same documentation, there's a section for the Deriva clients. In the Deriva clients, we have them for Macs, we have them for Windows machines, and also for Linux machines. So let's say we want to install it into a Mac. We'll have or a Windows machine, we have actual releases that you can download and install it like any other program. Or if you are using a Linux machine, you can directly do a pip install once you have Python installed in your system. So let's look at one of the releases, for example, for Mac. So basically, I see the Mac download. I basically download that file and just had to go through the installation, add all the defaults, and then in my Mac, what it will do is in my applications folder, it will create a folder called Deriva Client Tools, and over there I will see all these different utilities, the BDBAG tool that we showed earlier today, then there's an authentication tool, and the one we're going to look at now, which is the Deriva upload tool. So let's say I already went through the installation. So the next thing I'm going to do, I will go back to my computer and organize the files in the way that I need to. So I created here a directory where I already have the data. Let's say I call it the Bootcamp demo data. And now I need to start creating this structure using the real identifiers. So in this folder, I will call it with the RID from the dataset. So we go back to the dataset. I think the RID was this one. We'll copy, go back here, and rename this folder like this. Then I will enter the read for the replicate of the microCT mutant. So I will go back to my system here. microCT mutant. This was the read of the replicate. I will copy and name this folder like that. Then I will go to the microCT control. And rename the directory. And last, the RNA-seq assay, replicate ID. Okay, so I already entered all the actual RIDs, and then I have them already organized in the way I want it. So can you see now? Is it big enough?
Yeah,
So
yeah.
I have one directory here
Yeah.
for the data set, and then basically 3 directories, one for each replicate. And under the microCT replicates, I have an image IMG directory where I have the high resolution image. And then I have a thumbnail folder with the name of the image that this PMG file is the thumbnail of. Same thing for the other microCT replicate. And for the RNA-seq replicate, I have one folder for the process data with the corresponding mapping assembly, and then I have the bigwigs and BAM files. And for the sequencing data, I have the FASTQ files. So now I went through that part of organizing the data. So what I need to do is go to my applications and start the upload utility. So by default, the tool is already configured to point to the face-based machine, so you don't have to do any of that configuration. But the thing you need to do is you need to log in. So you log in using the same credentials that you use, to log into the system to create the data set, because you need a user that can add data to that project. So okay, so once I am logged in, the next thing I do is I click browse here and I navigate to that top level directory with the RID of the data set. Demo data. So I select the location with the RID for the data set. But once you go through that, basically the same way we have here the sequencing data, then we'll see also all the images and the thumbnails.
Yeah.
Actually, I can show you in this demo how it will look like. So we'll have basically the genome browser for the bigwig files, then we'll have the thumbnails for the images, and then we'll have the process data and the image data.
Yeah.
And we have some actually, for the imaging data, we actually have some very interesting, like what Rob already mentioned. We have this pipeline that once, let's say if you have already only uploaded the T files, then the pipeline will process the files and generate the thumbnails automatically for you. And same thing for if it were a single cell data, it will process the single cell data and create the visualization. You only have to upload the Seurat file.
Okay, so, I think it's pretty clear. I was looking for some demo on the website before, so this was very helpful.
Yeah,
through.
Yeah. Sorry about this. I don't know what went wrong. I think I'm having some files that were probably corrupted from a previous demonstration, but this tool actually works really well. And I don't want to tie you up now with trying to fix the problem, but trust us, it works really well. except
Yeah.
now. But
Something like that. Yeah. Something
um,
like that.
okay,
That's the
so
peril of live demos.
yes.
Yes, it is.
So I think that now is pretty much covers everything. So we basically created the whole data set and uploaded the files, and then you're done. Oh, actually, the only thing that is left of, so once you go through all this, you basically send us an email saying, okay, I'm done, I'm ready for review it, for you guys to review it, and then we review the information, and if everything's fine, we release it or we leave it in embargo state. Okay, so let me now go a very short description of what would happen if you were to upload the actual human subject data. So in that case, what you will do is you will create this, do this, just creating the skeleton of the as much as you can with experiment and the biosample that we talked before, and then you will probably just package all your data in some directory form, hopefully with the same structure that as much as we can as the one we've shown before. And then we will give you an encryption key. You basically have to compress that folder and encrypt it. We'll show you how to transfer to us in encrypted form, and we will place it in our dedicated server. And then if somebody later comes and requests that data, we're going to do the inverse mechanism. We're going to decrypt the data, package it for them, encrypt it with their own encryption key, and deliver the file, the data encrypted to them. But that's all communication we'll have to have. Once you're ready to submit the data, we'll explain you exactly how to. We actually have good documents on that tell you how to prepare the data. We'll help you on how to encrypt it and send it to us.
Okay, I think it's really pretty straightforward. And as I mentioned, I went through with my young colleague also on the website on demo, and this was helpful because you walk through
Right.
in kind of details and few things which I wasn't completely comfortable before. I think it's pretty much.
If you have, I can show you I can show you some examples of what I'm talking about. Let me try to find one that might be a good one. This one, for example, from Benedict Hargrimson is sort of a example like that. So you see, this is a human subject data for craniofacial morphometry. So they have a very large collection of like 3,000 or 4,000 or even more scans, right? So they have some basic information about the data set, and then we don't enter experiments and biosample for each individual in here because it didn't make sense. But what we have is we show a spreadsheet with all the identified information with some of the metadata that we make available to the public, and then so people can download it. And then we have all this data, all these scans stored in this secure server. So when people make a request, we deliver all that data.
Yeah, I can see that this is probably combined data from all these people who are listed here, which I do know personally. So this is maybe something where we can also contribute with our data, because we have some of them as well.
Sure. Yeah. So sorry that we kept you for so long and we bore you with all these very complicated things. But if you were to have a data set like this, it will be very simple. It's basically create a basic structure of the data set, enters information about the description and a few other terms, and then most of the part is going to be coordination with us on how to transfer that data.
Okay. Okay.
So it shouldn't be difficult or time consuming at all.
Yeah. Yeah. Very good.
So whenever you're ready, you just go here to submit, how to submit data, and fill in the data submission form, and that's basically where you start.
Okay. I'm so happy and glad you guys dedicated
Sorry.
all of you, your time to very private
Yeah.
lesson
Yes.
for 3 hours, 4 hours.
Sorry.
Yeah.
Right.
Yeah, thank you so much. It definitely was helpful, and then we'll be happy to get our data, contribute, and also most likely to use some data which you have in your databases, because it's a huge.
Yeah.
Yeah, absolutely. So if you have any questions, please email us at help@facebase.org. And we'll be happy to, when you have your specific requests, we can help you one-on-one again when you're ready to submit or to make a request.
Okay, definitely I will. And thank you again. And sorry that I wasn't on camera, because I like myself to seeing people and being with them. It's closest to the personal communication. But unfortunately, this is my old laptop, because my new one I left actually in another office. So I couldn't get my camera working on this one. Okay.
Oh, that's not a problem. Not a problem at all. But thank you so much for joining us today and spending your time with us.
Thank you. Thank you, guys.
Okay.
Absolutely.
Thank you so much.
Okay, have a great
we appreciate
day.
it. And
Thanks.
get in touch with us when you need any help.
I will. I definitely will. Okay.
Sounds
Thank
good.
you. Thank
Okay,
you
take
again,
care.
and have a good rest of the day.
Thank you, you too. Bye.